Good evening, everyone, and thank you for joining us for tonight's International Dermatology Education Foundation Educational Series webinar. My name is Ashley with RBC Consultants. Tonight's webinar is titled Exploring TYK2, a key player in the psoriasis IL-23, IL-17 pathway. As moderator this evening, we have Dr. Leon Kircik. He is president of the International Dermatology Education Foundation. He's clinical professor of dermatology at Icahn School of Medicine in Mount Sinai, New York, Indiana University Medical Center in Indianapolis, Indiana, and medical director of physician skin care, DermResearch, and skin sciences in Louisville, Kentucky. Our speaker this evening is Dr. Jason Hawks. He is a board-certified dermatologist coming to us from Sacramento, California. We would like to thank our supporter this evening, Bristol Myers Squibb, for making this educational event possible. Before we begin, a couple of logistic tips. If you're having issues hearing the webinar, you can listen to the presentation using your telephone. Just select phone call in the audio pane and the dial-in information will be displayed. If you're having any technical issues, you're welcome to use the questions tab on the right-hand side of your screen. At the end of the webinar, a survey will pop up in your browser and will be emailed to you within 1 to 2 days. We would greatly appreciate it if you would fill out this short survey. And also within 1 to 2 days of the webinar, a certificate of attendance will be emailed to you. So without further ado, I'd like to pass the floor virtually to Dr. Kircik.
Thank you, Ashley, and good evening, everyone. Thank you for joining us for another session of International Dermatology Education Foundation educational series. As you know now, we have been around for a while. Next slide, please. International Dermatology Education Foundation is a non-profit organization and whose principal mission is to raise awareness and improve dermatology care all over the world through education, especially in underserved areas. We have done originally live programs, and after COVID and during COVID, we started doing multiple webinars. As you see, we have been all over the world globally, in Canada, including North America, US, as well as South America and Europe and Southeast Asia. And we are really good at gathering the best of the best speakers in our programs. And tonight, we have one of the best speakers here in dermatology, rising young star, Dr. Jason Hawks, who I call him a good friend of mine, and he's gonna discuss the role of TYK2, exploring TYK2, I should say, in pathogenesis of psoriasis. Thank you and welcome to our webinar, Jason. Please take it over.
Thank you, Leon. I'm excited to present to the group tonight, and I think it's amazing that you have, so many attendees that come, and this is the new way of education where we can do this virtually. So let me just click here, and we'll start the presentation. So I'm going to be talking today. This is an unbranded talk by BMS, and this is exploring TYK2, a key player in the psoriasis IL-23, IL-17 pathway. And so by way of introduction for myself, I came from the science first before coming into dermatology. And this is a fun deck for me, probably one of the best presentations that I like to do, actually, because it brings the science back to the clinical stage. And I think this is a short deck. It's short and sweet, but it really gets to the key concepts. And my interest is really in looking at some of the novel immunology, some of the new pathways that is allowing us to manage disease better. No therapy is 100%, and we always are looking for these new pathways. So I'm excited to talk about this exciting pathway and really help you understand where TYK2 fits into the psoriasis immunology framework that we have. Right, so disclosure is the standard here. I'm a speaker of BMS. I'm a paid speaker. I did help put together this deck and create it for those of you that are listening. This has my disclosures here. Most of the work that I do is really consulting and speaking, really in the inflammatory disease space, and I don't own any stock in these companies, including BMS. All right, so what are we going to talk about today? One, I want to do a very high-level review of the overview of psoriasis. What's the initial framework the immunology that's happening in psoriasis, not related to any particular company or products, for example. Just want to sort of review that, point out a couple of predominant pathways we're going to talk about today. I'm then going to take you through that pathway and show you where TYK2 fits specifically in that immunology framework. And then the last point here, which I say is really the key point to this presentation, I really want you to understand how TYK2 is different from the other Janus kinase members in the family, specifically JAK1, 2, and 3. These often get lumped together. They shouldn't be lumped together. They have very different biological functions, and that pathway, its relevance to psoriasis is also very distinct. So that's going to be one of the key aspects that we're going to talk about through this presentation. All right, so we're going to start with the overview of psoriasis. And again, this is very high level. This is just our current working model of the immunology model in psoriasis or psoriatic disease. When we talk about psoriasis to set the stage here, we're really talking about a chronic inflammatory condition. This is a T-cell mediated disease. It's immune mediated. It's driving this chronic state of inflammation. Most of the patients that we see with psoriasis, as you know, have about 85% have plaque psoriasis. We know that it has predominant areas of involvement, such as the scalp, the elbows, the knees, the hands, the feet, the genital area. But we know psoriasis, like many of the inflammatory diseases, can present anywhere in the body. And when we start to look at the heterogeneity of this inflammatory disease, this kind of shows you some of the breakdown and proportion of where we really start to see this disease present in our patients. Now, as you know, in taking care of patients, we can see a combination. Some people only have scalp disease. Some might have entire body disease involving the scalp as well. So we want to kind of look at the variation of the presentation of our patients as we start to understand what we're going to do in sort of helping to mitigate this chronic inflammatory disease in our patients. And what's interesting about the immunology of these different body areas is we really don't have a good understanding of the nuances that drive a particular, endotype or phenotype of disease. For example, why do patients just get scalp disease? We don't have a very good understanding of that. But one of the things that's going to tie through in this presentation is that despite the clinical variations, the heterogeneity, we still see a consistent factor, which is that the IL-23 and 17 signaling axis or pathway is really predominant to this disease. And that's going to be really critical as we start to say, where does TYK2 play a role and how does it fit into that predominant signaling pathway that we hear about all the time, which really is the key driver of this inflammatory disease. And as this pathway gets activated in our psoriasis patients, it leads to systemic inflammation. So not only do we have skin inflammation, we start to see inflammation of other organ systems and other tissues. We see comorbid conditions such as cardiovascular disease, obesity, metabolic syndrome. We see psoriatic arthritis, so involvement of the tendons or ligaments and also the joint space. We see other disease associations which look dramatically different from skin presentations, such as inflammatory bowel disease. We see mental health, depression, anxiety, suicidal ideation. We know that just having disease, irrespective of our decisions to manage that disease, we see that systemic inflammation as driving some of these other factors, which is really why we're aggressive in trying to manage holistically our patients, not just the skin manifestations. And again, the relationship between the IL-23 and 17 pathway, we haven't worked out those nuances, but we know that it has some relationship there, and a lot of research happening in this exciting area, which is how can we tease out the immunology of some of these co-occurring diseases. And of course, as a clinician, what's most important to us is what's the impact of these diseases, not only on the health of our patients, but also on other aspects of life. So we start to look at the quality of life impact, the impact on productivity, intimate relationships, their mental health. We see this heavy burden that severe disease has on our patients, and even the impact that some of these very localized variants have on our patients. And two examples of that, well, actually three: predominant scalp involvement without other areas has a very high impact. We see patients who have genital disease and no other involvement in other body sites. We see a very a large quality of life impact and mental health impact there. Also hand and feet involvement. So variants like palmar plantar pustulosis, for example, we see this has a significant effect on our patients. And we want to address all of these aspects. Now, if there are any early career trainees or residents that are attending, I want to just point out one thing that was really a myth when I received training in psoriasis, which was that one of the differentiators from psoriasis versus eczema is that eczema itched and psoriasis didn't. And we know that that's just fundamentally untrue. Our patients with psoriasis can have predominant itch, so it's really not a discriminating factor from determining psoriasis versus other inflammatory conditions, say like atopic dermatitis or even prurigo nodularis, which has really predominant itch. So I want to just point that out, since we've really kind of changed course on some of the earlier didactic information that I had in training. So then if we shift gears and say we've got this clinical entity, we have multiple endotypes or different phenotypes which can present in our patients, what's happening under the skin that really can account for the vast majority of sort of the cases here? And I want to start with the IL-23 pathway, but I'm going to tell you on this slide, I want you to remember 3 key pathways, and that's going to be important for some of the later aspects of the presentation. So the first pathway is right here. We can see this link between IL-23 and IL-17. So what's happening in psoriasis skin is that in these lesional plaques, we have high levels of IL-17. And the key take-home point here is that IL-17 is predominantly what drives the phenotype of psoriasis. So when you see those indurated plaques, the erythematous plaques, the scaly plaques, that's predominantly driven by IL-17, not IL-23. IL-23 on the skin actually doesn't do a whole lot to the skin. So what's the role then of IL-23? So IL-23 is upstream of the T cell. And what that means is that it's a signal that's made by other cells like the dendritic cells here. And IL-23's primary purpose in psoriasis is to help differentiate T cells that make high levels of IL-17. So you can see here we have IL-23 made by another cell that's going to help these T cells differentiate into high-level IL-17 producing cells. And as they make IL-17, we're going to lead to the phenotype of psoriasis, which you can see here. Again, that keratinocyte proliferation, that erythema, that induration, and that scale formation. So the first pathway, which is by far the most predominant pathway, which is what you're most familiar with in choosing management options for our patients, is that IL-23 pathway. But there's 2 other key pathways we don't spend a lot of time talking about, and I want to just highlight these. So the first pathway, which is the second you're going to remember, is this type 1 interferon pathway. So these are inflammatory cytokines predominantly made by non-T cell types, or at least a unique subset of T cells called the innate lymphoid cells. You might hear of ILC3 cells. These are cells that don't predominantly make IL-17, for example, but they can make other cytokines such as the type 1 interferons. And these type 1 interferons are going to add to the complex immunology that's happening in the skin. And the other pathway is very analogous to IL-23, which is the IL-23 pathway, or the IL-12 pathway. The IL-12 pathway, like IL-23, it's a signal, it's a regulator of helping T cell differentiation for type 1 cells or Th1 cells. And the type 1 cells are going to make other cytokines like TNF alpha or interferon gamma. And as you already know, this can contribute to psoriasis. So how do these cytokines work together? They're going to work in conjunction with IL-17. So we have IL-17 predominantly driving disease, and then we're going to get these other cytokines that are going to work synergistically together with IL-17 to help drive more inflammation in the body. And so that's going to be the 3 key pathways. So IL-23 and 17, the type 1 interferons, and IL-12. And I'm going to show you where that's going to be relevant to how TYK2 works in psoriasis. So this is going to just highlight, again, that key pathway, because we're going to use this framework to show you where TYK2 fits in. So what do you see again, as we've just talked about? We have dendritic cells, for example, as the predominant source of IL-23. IL-23 is going to help the T cells differentiate into these type 17 cells, which are going to make high levels of IL-17. As we make high levels of IL-17, we're going to drive the plaque, right? So you can see a pretty classic plaque there on the knee, erythematous, scaly, thick, indurated, and that's going to be the predominant role for the IL-23 pathway. And then as the keratinocytes become activated, they start making signals of their own. These are things like IL-19, IL-36, IL-1, IL-22. These are going to be cytokines that the keratinocyte starts to make in response to high IL-17, and this is going to feed back to the dendritic cells. And this creates that cycle where it just keeps going, that feed-forward cycle. We sometimes call this feed-forward inflammation. So we get this predominant dendritic cell and T cell signaling. We start to make psoriasis. The skin becomes metabolically active, starts making a number of proteins and cytokines that are going to feed back to the beginning of that system, and that's going to create the cycle of chronic disease in plaque psoriasis. And that's true for the most of the subtypes in psoriasis, actually. And so then we say, where is TYK2 going to play a role in this predominant pathway? So we've got IL-17 and IL-23, and if that's predominant pathway, then why are we even talking about TYK2? And this gets into some really important concepts about how do these cytokines actually work in terms of affecting the cell. So there's a couple of things I'll highlight from this slide. We won't spend a lot of time on the history of psoriasis, but this really kind of shows you the relative discovery dates of key cytokines. And you can see in the early mid-90s here, TNF's discovered. We don't really learn about IL-23 until the early 2000s. Very shortly afterwards, we see that IL-23 also involves TYK2 as part of its signaling. And then we start to learn about IL-17. So this is kind of a nice way of remembering it, because with IL-23 and IL-17, the discovery of TYK2 is kind of sandwiched in between those. And that idea of it being sandwiched or in between IL-23 and 17 is going to help you remember what its normal function is. And then this last point here in 2010, when we're looking at what are some of the genetic causes of psoriasis or psoriatic disease, we discover that TYK2 as a gene can have susceptibility implications for psoriasis. So the type of mutation, whether you have a gain of function or a loss of function mutation, is going to affect our risk of psoriasis. And I'm going to click on this button here in the top right. And this is actually going to go through a really, I think, an interesting proof of concept of why TYK2 is important. So kind of going back to basic cell biology, we're talking about the types of mutations that can affect genes. And what we're going to talk about is two different concepts. One is a gain of function. A gain of function mutation is where a gene becomes more active. And that's in contrast to a loss of function mutation, where TYK2 is not functioning properly. And remember, we have two copies of each gene, so we can have one normal and one overactive, or one less active, or we can have two defective or inactive functions of a gene. And what this graph shows you is that when we look at TYK2, right here, we're looking at a heterozygous mutation or a variant. And these are patients that have one normal copy of TYK2 and one abnormal copy of TYK2. And what you find, so that TYK2, one copy is not working properly, we see a decreased risk of psoriasis. So this is an odds ratio, and so less than one is a decreased risk. So having one defective copy decreases your risk of psoriasis, but also some of these other inflammatory conditions. And now if we look at these patients that have this mutation but have two defective copies, we see a big difference in their risk. So you can see a very dramatic drop in the risk of psoriasis, as well as some of these other inflammatory conditions. So what does this tell us about TYK2? It tells us that for whatever the mechanism is, TYK2 must play a role in inflammation, because if you disrupt it genetically, just as we've shown here, you can decrease the risk of the inflammatory conditions. So that's what I'm going to try to put together for you, is that story, because this is a nice proof of concept before we start to tease out exactly how it works. So we're going to go back to the chronology here, and then I'm going to walk you through how exactly TYK2 reduces that inflammatory risk. But you also learned another aspect of this from the deck, which is that TYK2 must play a role not only in psoriasis, as we saw, but also some of these other inflammatory conditions. And one that you saw in that graph that's not listed here is multiple sclerosis. So what that means is that TYK2 must be playing some role in the immune function, the normal immune function, And interestingly, if it's impacted in a positive way where it's more overactive, we can increase the risk of these inflammatory diseases. Yet if we have a defective copy or it's not working, we can reduce the risk. So that tells us that whatever TYK2 is doing in the immune system must play a role in the pathogenesis of various and multiple inflammatory conditions. So let's look at that in plaque psoriasis, which is really kind of the focus today. So this is going to build on that framework that we just talked about, which is that IL-23 is going to have an impact on the T cell, which is going to result in increased IL-17 levels, which is going to drive psoriasis. And so you can see that here, high levels of IL-17, it's going to result in the formation of a plaque. So how does it work in terms of TYK2? Well, what we know about most cytokines is that they're large proteins. They don't get inside T cells. So how do we get a signal like IL-23 to tell the cell to make more IL-17 if it can't get inside to the nucleus, which is where all the genes are held? And so this is kind of a framework for most of the cytokines. So you can see IL-23 here. Now this template's going to work for virtually any other cytokine, but we're going to use IL-23 here since it's the focus. So we see IL-23 here. It's going to bind to the receptor on a T cell. You can see this is a T cell that's primed to make high levels of IL-17. IL-23 is going to bind to its receptor, and you can see on the inside we have TYK2 and JAK2. Now this is where the JAK-STAT signaling pathway comes in. So if you've learned about JAK-STAT signaling with a number of other products out there that you've heard about or read about, what we're really talking about is it's really the handoff. It's from the outside of the cell where the cytokines are, handing off to the JAK-STAT signaling pathway, which is an intracellular signaling pathway. And that's going to carry the message of the cytokine inside the cell, into the nucleus, and tell it what to do. In the case of IL-23, high levels of IL-23 are going to activate the IL-23 receptor. It's going to activate TYK2 and JAK2. They're going to work in pair. And together, they're going to then activate STAT. So that's the JAK-STAT pathway. It's going to go inside the cell, tell this cell to make high level of IL-17. And as we have that, we're going to drive psoriasis. So what's the key take home here, which is that you can't get from IL-23 to IL-17 without going through TYK2. So that should make sense as to why it would be of considerable interest in psoriatic disease, because if you don't have a properly functioning TYK2, you're not going to get IL-23 making high IL-17. So this key point down here is that TYK2 is integral to IL-23 signaling, and we can't make high levels of IL-17 under the regulation of IL-23 without TYK2. So it's going to be a very key point. Now remember when I told you to remember it's sandwiched in between IL-23 and IL-17? This is really what we're talking about. The outside of the cell with IL-23, we go through TYK2, and that's going to result in this change, which is going to drive high levels of IL-17. So that's a really key point that we're going to then carry through in this model. So this gives you another analogy or another way to remember it, is that we can kind of go through this domino effect, that when IL-23 signaling is elevated in the skin or in the body of our patients, it's going to interact with the IL-23 receptor. It's going to go through, in this case, TYK2 and JAK2. Other cytokines are going to use other pairs. It's going to then activate STAT, and that's going to go through IL-17. And that kind of gives you the domino effect of when we go from IL-23 to IL-17. So again, TYK2 right smack in the center of IL-23 signaling and IL-17. So then let's walk this through in an animation so you can kind of see it. So here's our IL-23 cytokine. It's going to bind to the IL-23 receptor, again on T cells, because this is a T cell mediated disease. We're going to activate the IL-23 receptor. You're going to see this come in. You can see the activation. It's then going to activate TYK2 and JAK2. Both of them are required for normal functioning of the IL-23 signaling pathway and the IL-23 receptor. And then it's going to activate the STAT proteins, which are going to come together. They dimerize. They're going to move to the inside of the cell. And as they move to the inside of the cell, they're going to tell this cell to make high levels of IL-17, which we know is going to drive the clinical phenotype of psoriasis. So IL-17 is predominantly the cytokine responsible for driving the keratinocyte hyperproliferation and that plaque formation. So again, IL-23 has to go through TYK2 to get to the T cell making more or increased levels of IL-17, which we know is the predominant feature of plaque psoriasis. So this kind of gives you that key pathway as to how the IL-23 and 17 signaling pathway is associated to TYK2. So it becomes an integral part or a necessary part for this normal signaling pathway, which is why it became a particular focus in psoriatic disease. So that kind of sets the stage for what is psoriasis, what do we know about psoriasis immunology. We talked about 3 key pathways, IL-23, IL-17, that's the first pathway. The second one we talked about was type 1 interferons. And the third one we talked about is IL-12 or the Th1 pathway. So now we're going to shift gears in how does TYK2 differ from the other Janus kinase enzymes, particularly JAK1, JAK2, and JAK3. And then at the end of this, we're going to bring it all together where TYK2 is going to bring all of those pieces we've talked about, the immunology, to make sense as to why it's of focus in psoriatic disease. So again, when we talk about the JAK family, we're talking about this particular family, the Janus kinase, that's the JAK family, 4 enzymes, TYK2, JAK1, JAK2, and JAK3. So what I really want to help you understand is what are the structural differences between these enzymes? What are the functional differences? Now, as a physician, it's the functional biological differences that play out in the clinic, right? So the structural differences are a little less important until we think about sort of disrupting these enzymes. The biological difference is what I really want you to understand tonight, because TYK2 has such a dramatic different biological function than JAK1, 2, and 3. So we're going to talk about 2 key things, structural differences, which I'm going to go through here. How is TYK2 different from JAK1, 2, and 3? How are they similar? How are they different? Functional differences, I'm going to really stress 1 key point that I want you to remember. One is that TYK2 of all of these 4 enzymes is the most restricted to the immune system. What do I mean by that? It means that the function of TYK2 predominantly plays a role in the immune response, and it doesn't really play a predominant role in any other key biological processes. And that's going to be in contrast to the JAK1, 2, and 3, which are going to have a lot of function in the immune system, but also they're going to have a lot of other biological functions. And this kind of creates what I kind of talk about as the immune collateral damage, right? Our goal for patients is how do we interrupt the thing driving their disease, but not impact normal biological processes which don't play a role in inflammatory disease. And that's going to be one of the key differences between TYK2 and the JAK1, 2, and 3 enzymes. So let's start first with some structural differences. So what you're going to see here is of the 4 members of the family, you can see that we have these animations. Now I'm going to explain the animation just briefly here, or this illustration. You see 2 parts for each of the enzymes. Now I don't want you to think that these proteins are 2 separate proteins. These are just one big protein structure. These areas that are colored, so the blue and then the different colors, they're just different important areas of the protein. We call these protein domains. So again, not 2 separate proteins, but 2 key areas that we're going to focus in on. So the first one that we're going to focus in on is the same color, the light blue, on the right side of the illustration for each of these. So this is what we call the active or ATP domain, and this is similar across all 4 of the enzymes. The role of this portion of the protein is that it binds ATP. So again, going back to biochemistry and cellular biology, a kinase's primary function is to phosphorylate something else. So remember the pathway we just talked about. When IL-23 binds to the receptor, it activates the receptor and the 2 JAK enzymes. Well, how does it do that? It does it by phosphorylation. And then when the enzymes activate STAT, the JAK-STAT pathway, it's also going to phosphorylate the STAT proteins. So these proteins, these kinases, need to be able to bind ATP in this active site because that's what their main function is. And the ATP is the phosphorus. The P in that is really what they're going to then attach to other things such as the STAT proteins. And so it makes sense that this portion of the protein structurally is identical. Now, it's not identical and it's 100%, but it has the same identical function, which is to bind ATP. So you'll notice that this is similar. It's an identical function, but there's there's differences in terms of the protein structure. But you can see that the binding site for each of these proteins is very, very similar. So that means something that would fit into that pocket is going to fit in each of the other enzymes' pockets to some degree, because it's a very, very close match, right? Not identical, but very, very close. And that's going to be important when you start thinking about some of the clinical implications of things that can bind into this pocket. Now, that's in contrast to this other portion of the protein, which is, very related to work being done by BMS, which is that we're talking about the regulatory or pseudokinase domain. And you'll notice that for each of these domains, they're very different. They have a completely different color, they have a different shape, and that's to highlight that they have a different protein structure. So whatever would fit in this triangular portion on TYK2, for example, isn't going to fit in JAK1, JAK2, and JAK3. So one of the advantages to the regulatory domain over the active domain is it's unique. So something that binds into that pocket, for example, isn't going to have an effect on JAK1, 2, and 3. Now, we call this selectivity. And so we're talking about the ability to get a unique portion of TYK2 that isn't going to be shared across JAK1, 2, and 3. So very important to understand the structural differences and similarities of these enzymes. And in some of the other lectures you can get from BMS, you're going to learn why that regulatory difference is really key to a lot of work that they're doing. So then let's shift gears to some of the functional differences. And this is what really plays out for our clinicians, because it starts to tell us a little bit about the function of these enzymes. So when we talk about the 4 enzymes, we're really talking about how are these 4 enzymes going to mediate the immune response of many cytokines. So what I'll tell you is the Janus kinase family, it really has about a function of mediating signals for about 50 cytokines. So when you start to look at the individual pairings, you start to see that how they're paired is going to determine which cytokines are basically going to play a role in certain diseases. So let's kind of go through that. We're going to spend a little bit of time here because we have time with the webinar, and I want to make sure that you really understand this, because there's a lot of confusion here. So basically, when we're talking about the combination of these JAK enzymes, when we have 2 enzymes, so we have 4 options, how we pair these different enzymes is going to determine which cytokine. So let's take TYK2, for example. Everything for TYK2 is on the left side. When we have TYK2 and it's paired with JAK1, that's basically going to mediate the signals for type 1 interferons. And that should be familiar to you, because we just talked about that as one of the key pathways in psoriasis, right? So when we have TYK2 with JAK1 and they're paired together, that's going to form the receptor that's going to mediate the signal for type 1 interferons. Now, if we switch out JAK1 and we switch in JAK2, we're going to form a different receptor. We're going to get TYK2, which is then combined with JAK2. That's going to mediate the signal that we've talked about before, which is IL-23. But you'll notice that IL-12 is also the same receptor, and that makes sense with a lot of similarities that we know about clinically with IL-12 and IL-23 being related. So the concept here is that which pairing we have of the 4 enzymes is going to determine which cytokine it's going to mediate. And that's going to be helpful, because when we start to make predictions, which of these enzymes is going to play the most predominant role in a specific disease, say like psoriasis or atopic dermatitis or alopecia areata or hidradenitis suppurativa, we can start to make some predictions. So then we can just carry this through. We say, all right, well, what if we pair JAK1 with JAK2? Well, we see different cytokines here. We see interferon gamma and IL-6. If we switch out JAK2 and put in JAK3, so we have JAK1, JAK3, we see a different set of enzymes or cytokines. IL-2, IL-4, IL-7, IL-9, IL-15, IL-21. These aren't a lot of cytokines that we frequently associate with disease states, but there are some that we recognize. So we know that interferon plays a role in things like atopic dermatitis or alopecia areata. We see IL-4, which we know plays a role in a lot of allergic or type 2 diseases, including atopic dermatitis, probably prurigo nodularis as well. And then we can pair these enzymes where they can form what we call homodimers. So everything before is a heterodimer because the pairing's not identical. But now we can pair a JAK2 with a JAK2, and we see a completely different set of cytokines. But this is very interesting when we look at JAK2, because what do we also see? We see a lot of hormone type signals. So we see leptin, prolactin, erythropoietin, the growth hormone, right? So now we have a very different function in the body that moves even beyond the inflammatory mediators that we typically associate with cytokines. So now we have JAK2 not only playing a role in things like IL-23 and IL-12, so here's our JAK2 there. We also see JAK2 playing a role in interferon gamma, IL-6. We then see all of these other signals, including some of the hormones here. So what's the key takeaway from this slide is that, one, you can use this as a template to say, can we start to make predictions about which JAK enzyme plays a role? And the answer is yes. And one of the key take home here is that when we look at the cytokines that is involved over here in the TYK2 pathways, that means in any pairing, TYK2 mediates certain cytokines, we see that type 1 interferons, IL-23, IL-12, all three pathways that I talked to you at the beginning play some role in psoriasis. Now, again, I don't want you to confuse this. I'm not saying type 1 interferon is the key driver of psoriasis, but we know that these type 1 interferons do play a role in the pathogenesis. But we know that it's the predominant role with IL-23. But the key takeaway here is that TYK2 is really restricted in its ability to impact cytokines that play a role in psoriasis, these three key pathways. And really, we don't see it playing a role in things like these hormones or some of these other broad-acting biological processes. So one key take-home, TYK2 tied to psoriasis cytokines and very limited. And the second point here is that when we look at the action of TYK2 in the body, we don't see this very broad action. We see actually a pretty restricted set of cytokines and all cytokines that play a role in psoriasis. So that's going to be a key takeaway. So you can kind of see here, we're really looking at cytokine production and immune response for the TYK2 pathways. On the flip side, when we're looking at JAK1, 2, and 3, we see that these enzymes are going to mediate a much broader set of immune signals and cytokines, and we're going to start to spill over into that concept of that immune collateral damage, where we're moving way beyond the pathways of psoriasis and we're moving into other biological pathways. And then a couple of final points on this. And again, I wanted to spend some time here because it's really key, is that I always get the question, well, if TYK2 gets disrupted, we can see that it would make sense to help with psoriasis. So again, if there was a mutation in TYK2 like we saw earlier, would it help with psoriasis? The answer is yes. Now, you could also say, well, what would happen if somehow JAK2 was disrupted or it had a mutation where its biological function wasn't working? Would that also help us in psoriasis? And the answer is yes, it would, because if you don't have JAK2, then you're not going to have IL-23 and IL-12 signaling. You're still going to get type 1 interferon because it involves JAK1 and TYK2. But in addition to helping with psoriasis, we're going to see the shift where if JAK2 is not working properly, now we can't dissociate the impact it's going to have on these other key pathways. So in addition to blocking IL-23 and IL-12, for example, when JAK2 is not functioning properly, we're going to start to see this impact on all of these other signals, some cytokines, some proteins, and other hormones, right? So that's the idea that we need to think very carefully about what's not working properly, and if that's going to work for a particular disease, or if we're going to end up hitting a bunch of other biological processes which are really not relevant to what we're trying to do in helping manage our patients. And then the last thing that I want to point out is that when we start to think about, this pathway outside of psoriasis, we see that these other enzymes here probably wouldn't do much in terms of working for psoriasis if, for example, they were disrupted. Because we know that interferon gamma doesn't predominantly drive psoriasis, and we know that these other cytokines aren't key cytokines driving disease. So again, very clear separation from TYK2 as being psoriasis associated, as opposed to JAK1, 2, and 3, which are going to be associated with many other inflammatory diseases that are not psoriasis. And now the same is also true for TYK2, is that if we have TYK2, we wouldn't predict it to have an impact on diseases like alopecia areata or atopic dermatitis, for example, because again, these are cytokines that don't really play a role in those diseases. So spend a lot of time there because I think this is probably the most important concept that helps us interpret a lot of the information from podium presentations to the literature to other presentations you'll hear about how these enzymes are playing a role in inflammatory diseases. So I'm going to show this to you in a different form because sometimes hearing this in a couple of different ways helps. We're not going to spend nearly as much time here. But what this is showing you again, what are the systems impacted by these select pairings? So again, pairings with the 4 enzymes, the JAK1, TYK2, and also JAK2 and 3. How are we going to see that pairing play out? Again, we're going to see the cytokines very clear in psoriasis with TYK2. But all of these cytokines are going to have some impact on the immune system. So what do you see is that all 4 of these have an impact on the immune system, which makes sense because you need all 4 enzymes to mediate these cytokine signals. And remember, I told you there's about 50 cytokines that are mediated by the Janus kinase pathway. So all of these are going to have an impact on the immune system. But herein lies the problem of TYK2 versus JAK1, 2, and 3, which is that we start to see things like JAK2, which is having very broad, non-specific functions, even beyond the immune system. So we see its impact on metabolic activity, lipid metabolism, bone or blood cell development. So again, remember erythropoietin that we talked about. So these aren't necessarily things that we want to disrupt in our patients with psoriasis because we don't really think of the blood system outside of producing immune cells as being responsible for driving psoriasis. And so what we see is that JAK2 is probably the broadest acting enzyme in the Janus kinase family. And then after that, we see JAK1, which is a little more narrow than JAK2. And then even more narrow than JAK1 is we start to see JAK3. So I think of JAK3 as covering very similar cytokines as JAK1. So you can see some overlap. You see IL-2, IL-2, IL-4, IL-4, IL-7, IL-7, right? So you're going to get the point where you can see a lot of the overlap that JAK3 has with JAK1. So one of the sort of developmental interests in JAK3 is that JAK3 kind of has the benefits of JAK1, but it's a little more restricted to the hematopoietic cell or the T cell or lymphocyte type population. So if that's one way that helps you remember JAK1 and JAK3, JAK1 a little broader, JAK3 a little more restricted to the immune cell and the hematopoietic lineage, JAK2 very, very broad acting, including hormones, and then TYK2 very restricted to those 3 key pathways that I talked to you about in sort of driving the pathogenesis of psoriasis. So that's a way to kind of help bring that concept home here. So this really helps us understand where TYK2 plays a role in the pathogenesis of psoriasis, because we see how critical this combination of TYK2 and JAK2 for IL-23 and IL-12 signaling, but also TYK2 and JAK1, which is that type 1 interferon signaling. So we see it as being really sandwiched in between. So again, the domino effect, IL-23 is going to bind to these T cells. It's going to go through TYK2, and that's going to result in that high production of IL-17, which is going to drive the clinical disease. And that helps you kind of remember the role that this plays. And we'll just kind of end here with a couple of summaries. Here are some key points and takeaways, things I want you to remember. One is psoriasis is a complex, heterogeneous inflammatory disease predominantly driven by the T cell. Our ability to understand what's happening in psoriasis is really predominantly focused on the IL-17, IL-23 pathway, because this is really the key driver. But we're starting to learn the nuances of other cytokines that play a role in these variants, such as IL-36 is being the predominant driver for generalized pustular psoriasis. So while we typically talk about IL-23 as a prominent pathway, we're starting to tease out some of these clinically different phenotypes, and we're going to start to see where some of these other pathways might play a role. But in general, IL-23/17 going to be really central. We've talked about where TYK2 is very central and integral to this pathway, because without a normal functioning TYK2, you don't have a normal functioning IL-23/17 pathway. So if TYK2 is not functioning, you're not going to get IL-23 regulated at high levels of IL-17, and that's going to be really important. And as we start to look at the JAK-STAT pathway and these, particularly the Janus kinase enzymes, we're starting to understand how they're similar. We talked about that similar active binding domain, or that ATP binding domain, and how they're different. Very specifically, the pseudokinase, or the regulatory domain, where we see they're very unique to each enzyme. So again, TYK2 has a portion of the protein that's very unique, and what would fit in that protein pocket there isn't going to fit necessarily in a pocket for JAK1, 2, and 3. A very key point in work that BMS is doing. And then the last point is, we talked a lot about how the pairing of these enzymes together are going to mediate the function of various cytokines. Very specifically, TYK2 is going to pair with JAK1 and JAK2 to mediate key signals in psoriasis, and all of the other pairings are going to be other aspects of our immune system, or the cytokines in other diseases, and particularly broad acting pathways that are beyond the immune system. So again, one of the key take-home points, TYK2 very restricted to the immune system and highly associated with cytokines that drive the pathogenesis of psoriasis, which make it a particular interest to us as clinicians taking care of patients with psoriasis. And how can you learn a little bit more about the TYK2 pathway? A little QR code here. Reach out, find out who your BMS contacts are, either in both the medical and also the commercial. Again, today we were talking unbranded, so we didn't have any mention of any particular therapies or medications. There are other talks and things you can learn about in terms of learning about the commercial aspects related to BMS. But again, this is really just to bring you back up on the science. And we hope that you found this interesting. And we're grateful again to Dr. Kircik in inviting me here to present this to you. And I hope this was informative. Thank you.
Thank you, Jason. That was really amazing. I think we are very new in TYK2 pathway in dermatology, and we all needed this training because, you know, it's confusing, and especially the perception that it's sort of in the same family with the JAK, but it is so much different than the JAKs. So I think this was such an eye-opening lecture, going to the basis of it and understanding the pathway and the mechanism of action and how it plays a role in psoriasis, right? I remember when I was a resident, well, 30 years ago, you know, we used to treat scalp psoriasis with, I never forget, Keralic gel, which was salicylic acid, right? Ester gel, which was tar, and then Lidex gel, which was a steroid. And now if you do that, probably you get sued because nobody's going to put that on their scalp. So we have come such a long way, and we're so lucky to be here at this day that we can treat our patients with so much in our armamentarium and new drugs. So I thank you so much. I thank our sponsor, Bristol Myers Squibb, to make this educational event available. I think we are all done tonight. Next slide, please. And next April 9, we have another program coming up, which is also on psoriasis, and we will see Dr. George Han and Dr. Mona Shariari, who are both my friends, great speaker, and they will discuss across all skin tones with Tremfya. I thank you so much, Jason. I wish everyone a good evening. Take care. Bye-bye. See you on the next program.
Thanks again. Bye-bye.